forward to yes, thank you. I'm looking forward to this symposium serving as a basis for continuing exchange between the two universities. Thank you very much.
Thank you. Thank you, Professor Ho-Bom Kwon, a Dean of SNUSD. And the next message will be from Eijiro Jimmy, Vice Dean from Kyushu University.
Hi, good afternoon, everyone. My name is Eijiro Jimmy. I'm professor and chairman of the OBT Research Center and the Vice Dean for International Affairs. Before speakers talk, I'd like to briefly introduce the two major research centers which speakers belong of Faculty of Dental Science. May I use the PowerPoint slide? Okay.
Yes, you can. You can share it right now.
You can see that?
Yes.
Slide?
Yeah.
Okay.
I can see that slide.
Oops. Since the establishment of the Faculty of Dental Science 2000, we have been working to enhance education, research, and dental care under the philosophy of the overall well-being through oral health. However, in Japan, which is a top runner in the super aging society, it is an important issue to extend the healthy life expectancy of the people. Therefore, in 2016, we established the OBT Research Center based on the concept that maintaining oral health leads to the brain health and total health. And I became a chairman in 2017. Furthermore, we have just established a new dental craniofacial development and regeneration DDR research center to promote basic research and clinical application for the realization of the regenerative medicine in oral region this April, with Professor Fukumoto as the chairman. This slide shows the concept of the OBT Research Center. As you know, in recent years, it has been reported that oral health is linked to brain and total health, such as relationship between tooth loss and dementia, and the prevention of aspiration pneumonia by oral care. The OBT Research Center comprehensively studies oral function, brain function, and total health. And we also selected young principal investigators from each field and support their promotion. This slide shows ongoing project at the OBT Research Center. Today, I don't mention about each project. 9 PIs, including me, belong to the OBT Research Center. Dr. Wu will give a talk today. In addition, 2 former members have already been promoted to associate professor and full professor at other universities. At the DDR Research Center, basic research on tooth development and tooth and periodontal tissue regeneration and the development of new dental materials, tissue regeneration and cell therapy using stem cells, and clinical research are conducted at the collaboration with Faculty of Medical Science, Center for Clinical Translational Research, and companies. And Dr. Yoshizaki will give a talk today. In addition, we manage data obtained from the research and the bioresources such as cells and animals. In the future, we will concentrate the research conducted at the Faculty of Dental Science into these 2 research centers and aim to realize overall well-being through oral health by these 2 wheels. If you have any question regarding OBT Research Center and the DDR Research Centers, please contact me from this email address. Thank you for your attention.
Thank you, thank you, thank you for your message and also the introduction to the research OBT Research Center. And let's move on to the next welcome message from Young-Suk Park, our Vice Associate Dean, please.
Good afternoon, and thank you very much for your splendid introduction of your research center, Dr. Eijiro Jimmy. My name is Professor Young-Suk Park, who is currently the Associate Dean of Academic Affairs. According to the Wikipedia, the Kyushu University was established in 1911, right? And is known as a member of former imperial university with a long history in Japan. And the Faculty of Dentistry was established in 1950 something, as I heard just before. And I heard in Korea, the university has long been very close to the city of Busan, because they are very close between Busan and Fukuoka, and that they have been friends for a long time, as far as I know. In fact, since I was born in Busan, I have a more special familiar feelings to you. From now on, for today's events, I will speak in Japanese with the aid of Google Translate. Please don't laugh at my poor Japanese, please. 私は九州大学を2018年12月19日に訪ねたことがあり、その時この中村学長もお会いに機会が折しもありました。実はこれはその年の10月にソウル大学の歯科大学院開催された International Conference for Future Dentistry に当時の小屋の清学長が訪問していただいて、素敵な行為も知ってくださって、インタビューも応じてくれたが縁になっていました。今年、九州大学とソウル大学がこのような形のジョイントシンポジウムを持ったことは大きなご運だと思っており、私たちのソウル歯科大学院と九州大学の歯科部がこれをうまく活用して今後実質的な協力と距離が行われることを希望しています。また早くコロナ19自体が鎮静されて以前のように韓国の最も近い隣接国日本との往来がより頻繁になったらと思って希望します。今日素晴らしいシンポジウムも期待します。Lastly, but not leastly, I will let you know our YouTube channel address by text, all of you. All friends from the Kyushu University are welcome to enjoy it for more information about our school. Thank you very much. どうもありがとうございました。
よかったね。日本語が上手でした。
Yeah, your Japanese is much better than mine.
Oh, thank you so much.
Okay. Okay, before we move on to the presentation, I'm Shin Jung, a director of the International Affairs and Seoul National University School of Dentistry, and I'm really pleased to host this event. And if you kindly turn on the videos and show your faces on the Zoom, this symposium, we could take a picture, actually a captured video, but we could take a picture, and this moment could be a good kind of a good memory, like 10 years later. So on a count of 3, if you turn on the videos, we could take a picture of each other. And okay, is there anyone who wants to join the picture? Okay. And before taking a picture, as you all know, we have we have 4 presenters, 2 from KYUSHU and 2 from Seoul National University. Okay, I think we're almost ready. So on a count of 3. 1, 2, 3. Oh, what? Sorry. 1, 2, 3. Once more. 1, 2, 3.
It may be the final call.
Captured. Oh. Okay. Oh, I think this is the one. I think we have all the. I think we got a good picture. Okay. Okay, let's move on to the presentation from KYUSHU University. The title for this presentation is Comprehensive Analysis for the Gene Functions Associated with Epithelial Mesenchymal Interactions in Tooth by Keigo Yoshizaki. Okay, Professor Yoshizaki, could you please?
The introduction. Can I share my slides?
Okay.
Oh, sorry. I need permission. Yeah.
Oh, really? Okay. I think we could, all of us could share the slides
Okay,
right
works.
now.
Can you see the slides?
Yeah, it's
Okay.
very visible.
So, okay, thank you for giving me an opportunity to talk here today. I'm Keigo Yoshizaki, the Orthodontics Department. Today's my topic is the comprehensive analysis for the gene functions associated with epithelial mesenchymal interactions in tooth. Epithelial mesenchymal interactions are critical for the various organ development, such as submandibular gland, hair, tooth, lung, and kidney. Recently, the techniques of tissue regeneration have been developed. And now we can technically create several organs by using combination culture method. However, the cell source is a big problem. Which cells can we use for the regenerative medicine? Previously, we reported that hairs are generated in a knockout tooth, which is mediator 1 transcriptional cofactor. This result indicates that the cell fate can be switched by the transcriptional regulation. These findings assumed me that these organs may be formed by some common mechanisms by using common genes. And later, the specific genes may determine the cell fate for each organs. To approach this hypothesis, we performed comprehensive analysis using the cage method. The cage is based on the cDNA sequencing technology. The cage can selectively sequence at the 5' end of cDNA using kerp trapper method. The cage specifically detects transcription start sites, TSS, of mRNA, including short RNA and non-coding RNAs. This is the comparison between the cage and RNA sequencing. The RNA-seq detects each exons like this. However, the 5' end is not clear. On the other hand, the cage represents very sharp peak at the TSS site like this. Cage method is fit for the transcriptional profiling. We dissected very early stage of organs, SMZ, hair, tooth, lung, and kidney. And as a control, whole embryo was used. The total RNA was isolated and then performed KYUSHU analysis. Finally, we found 127,000 TSS in our experiment. First, we make cluster dendrogram to confirm how the organs are genetically close. For this analysis, we use tissue development ontology group. As we expected, whole body is far from the other organs: lung, kidney, hair, tooth, and SMZ. Further, we found that the hair, tooth, and SMZ were relatively close in comparison with the other tissues. For further analysis, we performed heat map analysis. The red color represents highly expressed cluster in each tissues. Around here, the showing the commonly expressed TSS in the SMZ, tooth, and hair. This cluster is tooth specific, and this cluster is SMZ specific. These results indicate that the SMZ, tooth, and hairs are genetically close in TSS expression pattern. We also performed transcriptional profiling and then found that the 19 TSS are commonly expressed, and specifically expressed genes in each organs were identified. Next, we tested whether the two specific TSS were expressed in the epithelium or in the mesenchyme. As you can see here, the two specific TSS were mainly expressed in mesenchyme. We also confirmed SMZ specific genes are mainly expressed in the mesenchyme. On the other hand, the commonly expressed genes in tooth, SMZ, and hairs are expressed in the epithelium. These results indicate that the commonly expressing genes are epithelial genes, and organ-specific genes are mesenchymal genes. This finding suggests that the mesenchymal genes are potentially critical for the tissue fate determination. Then, we are focusing on the mesenchymal genes for tooth development. Here, we showed one example case for the analysis. Tooth-specific TSS. The lists are here, and we focused on this TSS on chromosome 15. We tested this TSS is how specific in comparison with the other tissues. We downloaded the datasets of several developing tissues from public database, and then compared with our KYUSHU data. This TSS is very specific in tooth in comparison with the other tissues. However, it potentially causes two different microRNAs, miR599 and miR875. So we tested which microRNA is expressed in tooth by quantitative RT-PCR. Then we identified miR875 is expressed We also identified the miR875 is expressed during early developmental stages in tooth, and it localized in dental papilla, which is mesenchyme. First, we analyzed how miR875 is regulated. Cage analysis detects the TSS accurately, so we searched potentially binding transcription factors by using Jasper database. Then we found that the homeobox transcription factor PRX1 and 2 may bind to the miR875 promoter region. The PRX1 and PRX2 expressed in early tooth developmental stage, showing similar expression pattern with miR875. We also confirmed the PRX1 and PRX2 upregulated miR875 expression in MDP cells. To test whether the PRX1 or 2 regulate the miR875, we performed luciferase assay. As you can see here, the PRX1 and 2 upregulate luciferase activity. We also identified PRX1 to binding motif is existed on miR875 promoter, which is TAATTA. So we constructed the mutant promoter and then tested. The mutant did not show the luciferase activity, suggesting that the PRX1 and 2 bind to miR875 promoter and regulate gene expression. So the next question is how miR875 regulate tooth germ development. At the early stage of epithelial mesenchymal interaction, the mesenchyme cells migrate and condense toward to epithelium to form interactions. To test this, we conducted migration assay. MDP cells transfected with miR875 were seeded into culture plate with a stopper placed onto the well. After the culture, the stopper was removed. Then dental epithelium were placed into the areas without cells. Here comes the result. The MDP cells transfected with miR875 migrate toward to the epithelium. Scratch assay showed same result. However, the cell proliferation did not change. This result indicates that miR875 may regulate mesenchymal cell condensation toward to dental epithelium during early tooth development. The Pdgf signaling is essential during embryonic development, and it is especially important that its ligand Pdgf-A is related to neural crest-derived cell migration. We tested the expression by RT-qPCR. Pdgf-A is highly expressed in dental epithelium. On the other hand, the receptor Pdgf-R-A is expressed in the mesenchyme. This result indicates that the Pdgf signaling pathway plays a role in epithelial-mesenchymal interaction in tooth development. Next, we tested whether the Pdgf signaling pathway is involved in MIR875 expressing cell migration. Pdgf-AA containing beads dramatically increased MIR875 expressing cell migration. And we confirmed that the MIR875 expressing cells were migrated toward to the Pdgf beads. We also examined the scratch assay and showed the same tendency. This result indicates that the MIR875 is a regulator for the epithelial-mesenchymal interaction, regulating Pdgf signaling pathway. So the next question is how MIR875 regulate cell migration. In Pdgf signaling pathway, the several pathways are already reported. It is known that the P10 is a regulator for the cell migration. So we examined the P10 expression by using RT-qPCR and Western blotting. As you can see here, the P10 was downregulated in MIR875 expressing cells in both mRNA and protein level. And we found that the MIR875 potentially bound to the P10 mRNA at 3' UTR region. So we make mutant construct, then check the luciferase activity. MIR875 inhibits the P10 luciferase activity, but the mutant did not change, resulting that the MIR875 can bind to P10 mRNA and it regulates the expression. We also tested the phosphorylation of AKT downstream of P10 and found that MIR875 induces AKT activation. These results indicate that the MIR875 may regulate cell migration through the P10 expression. We also explored the MIR875 target genes by using cage analysis. And we found that the STAT1 and also STAT2 is downregulated in MIR875 expressing cells. We also confirmed the STAT1 siRNA increased cell migration in MDP cells. STAT1 is also involved in the cell migration in Pdgf signaling pathway, suggesting that the MIR875 regulates the migration through the STAT1 expression. Finally, we tested whether the MIR875 affects tooth germ development by using organ culture method. Dissected E13 tooth germs were transfected with miR-875 inhibitor by the electroporation, and we cultured them for 7 days. As you can see here, the tooth size is small and the cusp formation is inhibited in the miR-875 inhibitor group. This result indicates that the miR-875 plays a role for tooth organogenesis. In this study, we identified a tooth-specific non-coding RNA, miR-875, by using comprehensive cage analysis. miR-875 is expressed in dental mesenchyme during early tooth development. PRX1 and 2 transcribed miR-875, and it regulates the P10 and STAT1 involving Pdgf signaling pathway. In summary, secreted Pdgf-AA from dental epithelium bind to Pdgf receptor in mesenchymal cells, and it induces cell migration. Under this condition, the miR-875 plays a role as a regulator for mesenchymal migration in epithelial-mesenchymal interaction. They are our team member in the orthodontics department. miR-875 study was performed by Dr. Funada for his PhD work. The cage analysis was performed by Dr. Miyazaki. I really appreciate to all of my collaborators. We are supported by these grants. Thank you for your attention.
Thank you, Professor Yoshizaki. Please, you could also use the chatting room. We have the chatting tool on the right side. If you press the button on the lower bottom, you could also raise your hand or turn on your video if you have questions for the presentation. We planned for 10 minutes, 20 minutes of talk presentation and 10 minutes of Q&A session. So if you have any questions, please turn on your videos and turn on your sounds.
Professor Yoshizaki, I have a question not directly in your research, but your research, your lab. I mean, would you introduce your lab for us? I mean, the faculty members or your number of the researchers or the equipment and then areas, something like that, please.
Okay, thank you. Thank you, the question. And I'm working on the orthodontics department. And now we generally doing the basic research using, and I'm very interested in the tooth development. And as I presented, I searched abundant genes which are maybe regulating the tooth development and also the cell fate determination. And now our team is focusing on a lot of genes, and now we are analyzing them.
Okay, you said that you are in the department of orthodontics. Are you an orthodontist, a clinician?
Yeah.
Oh, really?
Yes.
Quite amazing. Yeah. So you treat patients right now?
Yeah. Really.
Oh, okay.
Yes.
Can I state the portions of your treatment and research time per week, something like that? Would you tell us that how much time you assigned for treatment and then for research?
Yeah, usually half and half, but yeah, now I'm focusing, focusing in the research for it. Yeah.
Mm-hmm. Mm-hmm. Okay, thank you very much. Quite impressive. I think
Thank you.
your presentation will a lot of stimulus for us. Thank you.
Thank you.
And any other questions for the presentation? Okay. Okay. If we maybe if you have any questions after we move on to the next presentation, you could still use the chatting tool. So after we finish all the 4 presentations, we could also have another Q&A session. So please leave your message on the chatting tool, and we after we finish all the presentations, we could go over again. So thank you, Professor Yoshizaki, and please big applause to the presenter today. Thank you.
Thank you.
Thank you very much. And let's move on to the next topic from Professor Joe Wu. The involvement mechanism of Pdgf infection in Alzheimer's disease.
Okay.
Okay. Yes, I could see the slide. Good.
Okay, can you see the slides?
Yes, I do.
Okay, good afternoon, everyone. I'm Joe Wu. Today, I will introduce a topic of oral health and bone health. So like right here, because I'm from the OBC Center, so my topic will be on the oral health and brain health. Okay. So first of all, I want to share about information about this graph. So you can see here, so aging is a problem in all of the world. And I want to share about the speed of the aging of the population. So seeing here, the speed of aging population will get fasterly in our aging area, including Japan and Korea. So that will be like us recognize the speed of aging. And dementia, the topic of mine today, is the major cause of the disability among the older people all over the world. And another information is about that in the next 13 years, the number of people with dementia will be expect trouble. I mean, trouble, not double, it's trouble. So that it will be get a big problem in the world, especially in our aging area. So let's also review about the dementia. So dementia is a proximity neuron degenerative disease. So in dementia, they have several types around that. And the topic I will talk today is about Alzheimer type dementia, that including about 7% of all of the case. And another important information is about that. I'll close that. So out of the case of Alzheimer's disease, so the family, that means linked with gene, only including less than 5%. And most of the case, that means the over the 95% is symphonic type of Alzheimer's disease that is increasing with aging. So that will be linked with the out of the case of dementia that will linking with the Alzheimer's disease. So let me talk about the hypothesis of Alzheimer's disease. So because in the brain of Alzheimer's disease, so A-beta accumulation, that means made A-beta plaque in the brain, and another is about the neuron fibrate tangle is formation in the Alzheimer's disease. So people are thinking about A-beta will be like a cause for induced synapse dysfunction and then make a neuron death to make a brain shrink, then make a dementia. Another hallmark in the Alzheimer's disease is about microglia activation. So microglia is what? Microglia just like a macrophage in the brain. And after microglia activation, so that will be introduced a neural inflammation. That means inflammation in the brain, and then induce the synapse dysfunction, neuron death, and just like this. And recently here, the infection will be a hypothesis have been developing because they have several bacteria and virus can be found in the brain. That means this bacteria and virus can be infection to introduce neuron inflammation and then induce this phenotype in the brain. So until right now, the three hypotheses of pathology of AD has been established. And another concept is about system inflammation. That means how about the inflammation can induce aging-dependent neuron inflammation. And that's very interesting. We have the same level of system inflammation in the young adult. So neuron, so in the brain, microglia and other cells can be like a response to protect neuron. That means produce some protect factor to protect neuron. But in the case of either people only in the middle age adult. So after the similar system information come on, so microglia can be response to produce some factor can harm neuron. So that means the system inflammation can induce the aging-dependent neuron inflammation, and more seriously, even we have only in the middle age, so the response in the brain, how we will damage neuron. And periodontitis in our area, we know, is associated with a lot of system disease, just such as shown here. And people know there's the bacteria just linked to the periodontitis, just like the top related bacteria. The top is the P. gingivalis. And the P. gingivalis have some bacterial combinate, including gingiva, LPS, and femora. And people thinking about bacteria can be go through the circulation, that means into the blood, and can be infection of the organ or tissue. In the case of Alzheimer's disease linked with periodontitis, they have numerous of clinical evidence as we show there. So I did not read all of them, but the evidence to show clinical case just between the periodontitis and Alzheimer's disease. And another important thing is about the periodontitis, P. gingivalis, LPS, has been found in the brain of Alzheimer's disease. So that means the PG gingivitis can be into the brain during the life. However, the mechanism of PG gingivitis inverted in AD was unknown at that time. So depending on our acknowledge on the system between system and neuron inflammation, we just made a hypothesis of maybe P. gingivitis can be induced or promote AD pathology through directly induced neuron inflammation or enhance or induce the system inflammation. So we focus on some other organs like a spleen, like bone and liver. The first of all, we see what happened after the chronic system exposure to PG-LPS. What happened on mouse. So we use two kinds of mice, that young mice with aging like 2 months, and we use the middle-aged mice, just like aging with 12 months. So we inject PG-LPS every day and see what happened what will happen on the memory. So let's see the two figures. After 5 weeks injection continuously with LPS in the young mice, no memory can be go down. But in the middle age of mice, see here, the memory will come down. So that means memory decline can be induced in the even in the middle age mice. Then we see what happened in the brain of the mice. So we can see the brain tissue just from the hippocampus. So hippocampus is the region just related with memory, learning and memory. So we take out the tissue from hippocampus and see what happened here. So we use we focus on a kind of factor, just like IL-1 beta, and people know IL-1 beta can produce from pro-1 to metro-1. So the metro-1 will be released and induce the inflammation. So we focus on what happened on metro IL-1 beta use. So you can see here, during the time, so in the young mice, in the case of young mice, so IL beta cannot be increased. But in the middle-aged mice, the IL-1 beta can be produced. And most of the IL-1 beta produced just found in microglia, but not other will. That means system exposure to PG-LPS increase the metro IL-1 beta produced in microglia, only in the middle-aged mice. Then by our surprising, so in the middle-aged mice, we found it in the hippocampus, the A beta can be induced. That like me shocking. So that means after system exposure to PG-LPS, I induce the A beta in the neuron, just like here, like shown here. So that has thinking about that maybe PG-LPS can induce the phenotype of AD-like pathology. Then we want to see what will happen between microglia and the neuron. So I show here, we conduct a in vitro system. So we isolate the microglia and stimulate with PG-LPS and take the conditional medium of this and put them in the primer hippocampus neuron. So LPS is only the LPS stimulated to neuron, and MCM is used the conditional medium from microglia after PG-LPS stimulation. So you can see here, the A beta and the APP can be produced, but by our shocking, the exterior A beta, that means release A beta from neuron cannot be detected. But when we see in the neuron, so the A beta can be found in after applied with the MCM. So that means PG-LPS induce microglia dependent intracellular A beta accumulation in the neuron. So that means PGLPS induce microglia dependent AD phenotype in the brain. So then we just want to see what will be happen after gingivitis or LPS can be induced or apply system inflammation. First of all, we focus on spleen because spleen is the largest secondary immune organ for regulation inflammation, immune and inflammation response. So you can see here after PGLPS stimulation, so the spleen can be improved. And in the spleen, we can see the dendrocyte cell can be increased, and IL-6, another inflammatory mediator, can be enhanced, and IL-17, that is kind of an inflammatory CD4 T cell, can be produced. That means system exposure to PGLPS can induce the inflammation in spleen. Then we focus on bone. What happened on bone? We use the same method to exposure PGLPS and see what happened on tibia. That is long bone. It's not the bone in our oral area. We will focus on long bone. So we can see IL-17 as well IL-6 can be increased in the bone tissue. And very interesting, the increase of the IL-17 or IL-6 can be related with the bone volume. So that means system exposure to LPS can induce the inflammation in the bone or positively correlated with the bone loss in the long bone. Then we focus on another organ, just like a liver. So in the case of liver, we use the P. gingivitis infection. So not PG gingivitis LPS, we use the P. gingivitis infection with the mice. And we use the middle-aged mice. So you can see here, after we injection with the P. gingivitis for 3 weeks, so we can see the inflammation can be induced in the liver, mostly in the macrophage. And we also, so that is also by our shocking. So A beta can be produced in the macrophage of the liver. That means after infection with the PG gingivitis. And then we want to confirm what will be happen is really macrophagy can be produce A beta or not. So we directly stimulate with P. gingivitis on the microglia, not microglia, on the macrophagy. So you can see here, so A beta can be found in here. That means A beta can be produced by the macrophagy. And also when we use the tissue from the patient with a periodontitis, so we also can find A beta just look in the macrophagy in the gingivitis tissue from periodontitis patients. So that means P. gingivitis infection can be induced, the A beta produced from the inflammatory macrophage. So because P. gingivitis can be induced the system inflammation in spleen, in bone and liver, and also increase the pool A beta, out of the brain. So that's like us thinking, if this proliferate A beta can be influenced in brain or not. So then we focus on this issue. So we focus on the one kind of a receptor, just like a RAG, receptor for advanced gene condition and the production. Because this receptor is a receptor for A beta transported from periphery blood to brain. That means it's kind of a beta influx receptor. So that can be published on Nature in 2003. So we know this receptor can be as a receptor of A beta influx. So we see the mice after P. gingivitis infection, and we focus on the cortex because cortex is another reason for the memory memory. So you can see here, we focus on the brain-bone interface cell. This is a kind of intercellular cell. So the expression of ROD can be enhanced on the intercellular cell. So that means after P. gingivalis infection, so the influx receptor can be enhanced. Then we focus on the what happened on A beta, just around the vessel. So you can see here, we can see A beta can be induced in the brain, most of them around the cerebral endocell. And that is very interesting. The learning and memory decline is getting faster. I mean, after 3 weeks infection with PG gingivalis. So as me introduced previously, if we injection with a P. gingivalis, so the memory decline can be induced after 5 weeks injection. But in the case of a P. gingivalis infection, so the memory decline just occurred after 3 weeks. So that means P. gingivalis have more damage on memory decline than PG-LPS. Then we just conduct an in vitro system to see if the PG gingivalis can be like the A beta transporter is really linked with ROD or not. So we use this system to see what happened on the cell. So you can see here, after P. gingivalis infection, so the A beta just goes through the transport will be increased. And when we use the treatment with the inhibitor of ROD, so the A beta transporter can be reduced. So that means P. gingivalis infection induced the ROD-dependent A beta transporter through the endocellular cell. So that means P. gingivalis promotion the proliferate A beta increase in the brain through the, the interface between blood and brain. So in conclusion, after P. gingivalis and LPS can induce the A beta phenotype in the brain, and P. gingivalis LPS can induce all amplified system inflammation. P. gingivalis can induce the proliferate A beta pool, and finally, P. gingivalis promotion the proliferate A beta inflows into the brain. That means P. gingivalis can induce or promotion AD in multiple directions. So our research is ongoing. So today I just get a image. So one of our publication is acceptable. So we need only one push to get it accepted. And our research can be attract publisher in the Japan. So we got some newspaper and radio and also on television. So that means people is very interested on oral health and brain health. Finally, I would want to take a home message for all the people, because people know the A beta plaque can be occurred before the A beta onset, 25 years before the AD onset. And also the AD is a neuron degeneration, irreversible progressive disease. And more important is now in fact treatment has been developed right now. So if the oral care, like a treatment or prevention, may be as a respectable preventive approach for delaying onset and pathology process of AD. So please take the message, like a clean our oral clean, slow aging of brain and contribute to well-being. Thank you for your attention.
Thank you, Professor Wu. Thank you for the presentation. Okay, if you have a question, please turn on your audio and video. If you have a question.
Can I ask a question?
Yeah,
Professor?
please.
Thank you very much for your wonderful talk. I take your message very clearly.
Thank
So
you.
I try to keep my oral health at the best condition to prevent
Thank you.
Alzheimer in the future.
Thank
Well,
you.
yeah. My first question is that I'm not a neurobiologist, so my knowledge is very limited. Is there a consensus in the field that the peripheral source of A beta is more important than the A beta production in the brain on site?
I think because the pathology hallmark is A beta in brain, so people thinking A beta in brain is a source of pathology A beta. But we finding after Pdgf infection, so the A beta can produce out of the brain. And also they can have a possibility to let the peripheral A beta influence into the brain. So our body have a whole of the weight, so brain just only have a 2%. If 98% of the tissue can produce A beta to influence the brain, what happens in our brain? So that will be a big problem.
So that's very
it's serious. It's
yeah,
very serious.
astonishing. Actually, it's astonishing.
That means oral clean is very important for our brain.
But I wonder if there's data that shows that peripheral A beta is more significant than brain A beta.
Yeah, so we have published for several papers, but in this talk, I only pick up, only have the point by point. So like people understand the concept of that. So I did not talk about detail because today I think I will introduce 5 or 6 papers around our project. So it's a long project for over 10 something years, 17 years.
7, 8 years.
Yeah. So it's a long project.
Oh, I see. Okay. My second question is, you showed the rage expression, right? Is there any difference between young and old mice?
We did not see the young mice because young mice have no memory decline anymore. So in this time, we now focus on young mice. I think there will be something happen. I believe in something happen. But because you young mice have no memory decline, so I did not care. I did not care about the young mice. It's not good expression, But it's more important to focus on what we have happen on pathology, phenotype.
Thank you.
Thank you for your question.
I have a question.
Yes, please.
Oh, yeah. First of all, thank you for the interesting presentation, and it was really fun. And I have actually 2 questions. The first one is that is there any difference between the strains of Pdgf about inducing the Alzheimer's disease?
Oh, it's very nice question. I have many times to be asked this question. I only use one kind of strain. I did not test other strain, but by the paper, by other published, they compare with the strain. Something happened about it. So in our technician, we only can use one kind of strain. So I believe if you try to compare other strain, something will happen, and maybe other strain will have the same phenotype. I'm not sure, but I did not do that. I was always be asked by this question when I just submit a paper. So compare other students something. So I have no definition right now, so it's difficult for me. But some people do that. How about do that?
Okay. Okay, thank you.
Yeah, how about do that?
Yeah, I should try that. Also, my second question is that if the Pdgf is in the biofilm form, then does it have some difference in inducing Alzheimer's or like degree of Alzheimer's or degree of virulency, like affecting the Alzheimer's disease? Like, because usually Pdgf is in the biofilm form and it's
Very
kind
good
of
question.
trouble.
You're
Yeah.
smart. You're really, really smart. Very nice
Thanks.
question. Because you know, Pdgf only can be living in the low oxidation saturation.
Right.
So at that paper I will show, published by the 2013. So in that paper, they show Pdgf LPS of a Pdgf. It's not Pdgf because Pdgf I don't believe we can living a long time in the brain, but LPS and gingipan. So another group, we have a competition in US. So another group just focus on gingipan. So they publish on Science Advance and publish a lot of paper just on big journal. So they focus on gingipan because they think gingipan is a kind of enzyme can be make a more damage. So in my case, I just thinking about the long time, just have a very weakly damage can be make a big problem. But in their group, they focus on gingipan. So I believe other component will be have some phenotype because like I have induced. So when we injection with a Pdgf itself, so can be reduce the memory shortly than use PGLPS. That means other component will be work.
Oh, that's interesting. Thank you for answering my question. Thank you.
You're welcome.
And any other questions? Okay. Okay. Thank you for Professor Wu. And we'll after as I already told you, after all the presentation, we will have a little bit of time for the Q&A session once more. So please use the chatting room. And thank you very much, Professor Wu.
Thank you.
It was very interesting.
Thank you.
Thank you very much. Okay, let's move on to the next topic from Professor Shin-Young Park. The topic is U-polio-based learning and teaching in students' clinic. Professor Park, I can see the slide very well. Thank you.
Thank you. Can you hear me?
Yes.
Yes, I will start. Good afternoon, ladies and gentlemen. I am Shin-Young Park, Assistant Professor for Clinical Dental Education in Seoul National University Dental School. At first, I'd like to express my gratitude for having me in this joint symposium. Today, my topic is Electronic Portfolio System for Dental Education. I am a pediodontist, not an educational professional. However, when working in student clinic, I felt the portfolio assessment system is required for workplace-based assessment in clinical education, and I participated in development of electronic portfolio system. So here today, I'd like to share the concerns related to the portfolio system and introduce the portfolio system of our school. Before getting to the main issue, I'd like to discuss about the internal and external environment related to dentistry. Now, we are in Industry 4.0, featuring of cyber physical systems, smart working using cloud system, and artificial intelligence since 2015. This Industry 4.0 is quite different from the past. And in Industry 3.0, massive customization and automated production systems are important issues. However, Industry Industry 4.0 is focused on a personalized and precision production. And also, Industry 4.0 characterizes connectivity and openness through internet. So the information related to the medical and dental field is also enormously growing from day to day. With a flood of information, what kind of dentist are we going to develop for the future, dealing with uncertainty in decision making in clinical situations? In this era, we are needed to educate the students, have competencies to frame the appropriate clinical questions, to retrieve information, and to evaluate and weight evidence to make decisions, to draw a right answer in daily clinical practices. Similarly, the World Economic Forum defined that complex problem solving and critical thinking are ranked in top 10 skills required for the future relevant. So the most appropriate method to cultivating these skills is thought to be the portfolio assessment. Sorry. Assessment and evaluation is quite different. Assessment and evaluation is quite different. The purpose of assessment is to increase quality, not to judge it. Therefore, assessments have features which are going, positive, and individualized. In addition, through assessment, students are provided feedback based on evidence-driven measurements and measurement. And then students endeavor to achieve a certain level of competency. This is the famous Miller's pyramid of assessment. For the comprehensive assessment of competency, we need to assess knowledge, skills, attitudes, integrity in the real clinical practice. The portfolio assessment system is the method to assess this comprehensive understanding of students. Portfolio is a purposeful collection of evidences to demonstrate that learning has taken place, annotated with the student's reflections on what has been learned in terms of learning outcomes. The content of portfolio is personal development plan and monitoring, assessment, and reflection. The learning portfolio is a combination of these three contents. Portfolio is developed by collection of learning outcomes, selection of materials appropriate for purpose, reflection of their learning, and projection their materials to the standard. After that, portfolio is presented to the authorities concerned. For the purposeful learning and practice using portfolio, these components such as well-defined specific goals, planning, focus on task, feedback, monitor their progress, and motivation should be considered. These components are also corresponding to self-regulated learning, which is key paradigm of the modern education and also essential skills for Industry 4.0. Therefore, portfolio assessment is called to be formative and summative, qualitative and quantitative, personalized and standardized, and authentic assessment. The component of portfolio for medical and dental education is suggested by University of Dundee Medical School. In the core, performance of task, doing the right thing is assessed, and then approach to task, doing the thing right, and then professionalism. The right person doing it are assessed using portfolio. In detail, for the assessment of what the doctor is able to do, clinical skills, practical procedures, investigating a patient, patient management, health promotion, and disease prevention, communication skills, and information handling should be examined. And then, for the assessment of how the doctor approaches to the task, understanding of basic clinical and social sciences, appropriate attitudes, ethical understanding, and legal responsibilities, and appropriate decision-making, clinical reasoning, and judgment should be examined. For the professionalism, the role of the doctor within the health service and aptitude for the personal development should be evaluated in portfolio. For the process of portfolio assessment, students collect evidence of learning, clinical practice experiences, and then next step, students reflect their learning outcomes. More than 2 examiners evaluated the evidence and reflections. And therefore, interview the faculty, interview the students about evidences and reflections. After final stage, examiners finalize their assessment, such as pass, distinction, referred for remedy. For the successful introduction of portfolio in education, lots of factors are related. The support of academic leadership is one of the essential. Our academic leadership is fully understood the purpose of portfolio assessment in clinical education and support the development and management of portfolio assessment system. And next, all the people related to education, such as students, faculties, and dental education office are needed to understand the purpose of portfolio. And lastly, is thought that infra for the portfolio is affecting the sustainability of portfolio. Although lots of educators agree to the advantage of portfolio assessment system, lots of paperwork and time-consuming process are obstacles to need to overcome. So our dental school is developed. Our dental school developed an electronic portfolio system appropriate for our dental school. So workplace-based assessment in the clinical practice environment is one of the essential requirements of our system. In this context, we developed ubiquitous device-based portfolio system with company named Lab, professional developer for portfolio system for medical education. In this portfolio system, the role of mobile and personal computer is separately applied. Mobile phone is applied in dental clinics for workplace-based assessment. PC version of portfolio is applied for reflection and projection to standard. Through this ubiquitous system, we can apply the interactive learning in clinical education. For example, a student perform the clinical practice in student clinic, and faculty evaluate the performance during the practice and provide the appropriate feedback using mobile device. After practice, students reflect their clinical practice at home and study about questions related to practice. Faculties monitor their learning and reflection and can provide feedback online. This is the portfolio assessment model of our school. Students fulfill their portfolio about their learning, such as dental knowledge, patient care, clinical practice and observation, integrated clinical clerkship, self-directed learning, and professionalism. project analysis. At its phase, students' portfolio are reviewed by both faculty and students in the middle of the term. Faculty provide personalized mentoring for each student before final exam assessment. Until the end of the phase, students continue their learning with the mentoring results in mind. At the end of the term, faculty do final assessment based on their improvement of competency. Portfolio system of our school have 3 phases according to the curriculum. For the future, the portfolio system can be extending for the specialty training. Our e-portfolio system for phase 1, pre-dental course, students study basic behavior science and participate in projects such as public health and student research. So the portfolio system is fully supporting their collection of evidence and learning, such as reading materials, GPA, outreach program, certifications, language, and CPR certification, leadership programs, student research programs, such things. In the pre-doctoral phase, clinical practices, including patient-centered portfolio, clinical competence evaluation tests such as OSCE and CPX, 360-degree evaluation can be recorded. One of our featured clinical education system is longitudinal integrated clock sheet. It's also recorded by electronic portfolio system. Let's see our portfolio. I'm sorry that the portfolio is written in Korean. I'm asking for your understanding. In our school, longitudinal integrated clock shift takes place over a 12-month period. Students follow a patient from the initial visit and participate in each procedures in the various specialty clinics. Most of the specialty clinics in Seoul National University Dental Hospital involved in this longitudinal integrated clock shift, and students followed minimum 40 patients for 1 year. Students recorded every visit of patients which students participated in the portfolio system. After close of follow-up, students reflected their learning during the procedures and procedures. Faculty monitor their students' learning and assess the pass or fail and provided appropriate feedback about the case. This is the individualized monitoring system. For the well-defined goals and monitoring of a student's learning, we have visualized the system like this and tables to present their clinical practices and experiences. For the clinical competency of OSCE, objective structural clinical examination and clinical performance examinations can be tested and recorded in the portfolio system, and which are helping students planning and monitoring their development. In addition, the portfolio system provides students individual competence level analysis based on the data of the test and clinical practices. Using the personalized learning data, we can make learning analytics and personal plans can be provided in the future. For the faculty and school, the portfolio system also provides statistics such as feedback in class level and institutional level. In addition to student record, educational data can be obtained through electronic portfolio system. During collection of learning, educational data such as log data, number of clinical practices, quantity of feedback, timing of feedback, et al. are also additionally collected, and we can use these data for improving our educational program. For the improvement of our education program, we need to make decisions in evidence-based way, not by empirical way. E-portfolio system can be one of the resources for the institutional research, and we can progress the entire education system of our dental school. In conclusion, Seoul National University Dental Hospital adopted portfolio assessment for clinical dental education and developed the electronic portfolio system. This system is supporting students' regulated learning and interactive learning with faculties online. In addition, big data from electronic portfolio is essential resources for the institutional research for the educational program evaluation. Thank you for your attention.
Thank you for the presentation, Professor Park. And if you have a question, please.
May I have a question?
Yes, please.
Yeah. So, thank you for a nice presentation, a nice review for the half part of the presentation. And last, you presented a longitudinal integrated clerkship evaluation system for by electronic base, right? And actually, there are the when it comes to the evaluation, you have to use some sort of requirement, using a requirement to pass or not pass, right? But you also presented a kind of a personalized assessment for the individual students. What's the difference between these issues?
Thank you for the question. At first, our longitudinal integrated clerkship is a requirement for all the people, all the students, and but the case is very quite different from each student. So sure. we can have the we can provide feedback individualized based, because the student to reflect the cases according to based on the each cases. The cases are quite different. So some of the cases is the combination of the periodontal and prosthetic cases. Some of the others are based on the pedodontics and also such as those things. So this longitudinal integrated clerkship is very important, because the full follow-up is by the procedure, and they can make a vision to the improvement of the patient, and and all the procedures and in the text is just knowledge, but in various conditions they can be applied in different way and in the more modified way. So the people, students need to study in that way. But the type of the student clerkship is based on the evaluated by pass or fail, or some of the cases are pass or fail, some the others but each visit of the patient is recorded by the rubric evaluation form, because the according to the level of participation of practice, can student take the cases in some practice, and some in the others they can only just observation of the entire progress in specialty clinic. So we can make each evaluation in every visit, but all longitudinal clerkship is just to all kind of a combination and integration of all the cases, so it can just only pass or fail system.
Yeah, thank you. And you maybe you evaluate the student's ability using some sort of a combination of the clinical cases into a competency level, right?
Yes.
Yeah. How did you put the kind of milestone in each year? So using a competency or the a number of cases and kind of a experience of clinical cases or something like that.
We have some milestone of each year, but I think we have more definite milestone in the phase according to phase system, not the every year, because the first and second year is on. We have integrated curriculum, so basic and clinical science is combined. So we just only the discretely define the each milestone. We have instead before the clinical step, we have some milestone to pass the clinical competency before meet the real patient. And so and we have some number of requirements of the real patient evaluation, and so patient centered and we and after fulfill the requirement of the clinical practice in the real clinic, real patient, we can a student can progress the final examination of their competency for the graduation, and after that they can go to the they can take the exam for the national license examination.
Thank you.
Thank you.
I think the Professor Tsukiyama mentioned a very important point that based on what you said, that I think that the evaluation of the students has to be done by the qualitative test, not by quantitative test. But actually, up until now, our system, of course, we evaluate the competencies of the students, but we evaluate their quantity about how many cases they did. So I'd like to ask about some of the professors in KYUSHU University. What kind of system do you have right now?
We don't have such a very nice system in Seoul National University, but we introduced the e-portfolio system. It's commercial based. Each clinical cases, they put in the cloud system, and the instructor admit that which is good or not, like that. And we finally we have a kind of final exam, kind of a common test in the 29 dental school in Japan. Next from this year we have, and after taking two type of tests, when they pass and when they clear the minimum requirement, and they can pass the clinical practice session. So we only have e-portfolios, e-logbook system, only that, only the cases, number of cases, but not in the individual cases. The kind of portfolio is dependent on the individual clinical issues, like oral surgery or prosthodontics, perio or endo, like that. But we don't have any integrated portfolio system so far. Yeah.
Okay, I think the education can be developed more by way of competition and then research of other schools. So I hope that someday we'll investigate the system of the KYUSHU University. Okay, and then yeah, it will contribute to the education of the both universities. Thank you very much.
Thank you very much.
I think.
Yeah, thank you for your very good suggestion. So, Professor Tsukiyama is a specialist for dental education in this dental school. So I have one question. So I'm interested in your e-portfolio system so much. We are absolutely behind. So we must follow you. But so you established this system by your own. So you have many dental school. How about other dental school in Korea? They have their own system?
We have 11 dental school in Korea, and Seoul University and Yonsei. The Yonsei University use the portfolio system. At the yes, this system is the prototype of this system is developed in the Yonsei Dental School, and we adopted this system and we developed for our modified and developed for our system, and so we provide Yonsei University is our longitudinal clock ship systems and such more modification type. So we can now Korea Yonsei and Seoul National University use this system, and I heard that this company is preparing for the global version of the system, electronic portfolio system for dental education. So I think you can use this system also.
Oh, really? Thanks so much.
Yeah, Professor Shin Young-ba was in charge of developing the system in our school. And then, yeah, we are now in the stage of developing more. So yeah, I think the cooperation will help us and you a lot. Thank you.
Yeah, we should do that. Yeah. Thanks so much.
Okay, any more questions?
Can I have a question?
Yes, Professor Jimmy.
So how many students in each grade in your university?
How many students in here?
Yeah, students, yeah.
We have 80 or 90 students on year. A year. Yes, we are very, we have many students, and so we have 2 professors for the clinical education. So we have some responsible for the half of the students, each professor, and so we can make consult and mentoring and any other education for the professionals such as ethical problems in the student clinic. So yes, I think it's kind of we are some supervising for all. Yeah, so I think.
So one more question is for promotion, the student is the 2nd grade or 3rd grade. So what's the percentage of the examination on these portfolios?
For the phase 1, for the promotion, phase 1, from phase 1 to phase 2, they should certify or the kind of the certifications such as language, the CPR certifications, and outreach programs such as things. So they the portfolio assessment will occur, and if they all the requirement is fulfilled, and they can pass the another phase. But if they do not fulfill their requirements, some of the very minor problem, they can revise the portfolio for some 1 or 2 months. But the lots of the certificate is not can be fulfilled within 1 or 2 months. They cannot promote to phase 2. So just they should do the fulfilled all the things for the promotion of the phase 2 or 3. Yeah.
Thank you very much. You're nice talk. So we are very interested in your wonderful system. Thank you very much.
Thank you very much.
Can I ask one more question to the KYUSHU University? And then how many students do you have per year? And then how many clinical education is done? So I mean, which grade clinical education starts?
We have 53 students are here.
Okay. Yeah. And when the clinical education start?
When five, I
Fifth
think
grade.
5 days. Yes,
Yeah.
5.
Fifth and sixth.
Yes.
You have, okay.
Yeah.
Okay. Thank you very much.
So you might know the pass rate of Japanese national examination to get a license for dentist is so low, under 70%. But in the case of Kyushu University, 80%. How about pass rate in Korea?
About as far as I know, the over 95.
Over 95, yeah, over 95.
Over 95, yeah.
Almost all students are, passed their exam.
Yeah, we used to be that, yeah. But now, so examination is so difficult. So we have to focus on the pass rate. So we must focus to pass the examination rather than to educate good dentists. That's our problem. But we should follow your system. Yes. Thanks so much.
In fact, we're going to introduce the skill test as a national examination since this year. So our pass rate, I guess, will be lowered in the near future.
Thank you.
Okay, thank you very much for the presentation and all the questions. And let's move on to the last topic. Our last topic is digital workflow for planning, simulation, and 3D printing in orthodontic surgery, presented by Hunju Yang. Professor, thank you. Professor Yang, are you available? Yes, I can see the slide very well.
Hello, Hello, professors and colleagues at KYUSHU University and Seoul National University. It's a great honor for me to have the opportunity to present at the joint symposium. My name is Hunju Yang from Department of Oral and Maxillofacial Surgery, and I'm majoring in maxillofacial deformity. Today, I'd like to share my short experience with digital works in orthodontic surgery. From this phone I saw at my grandma's house when I was a child, to the smartphone that I carry in my hand every day, over the past 30 years, communication methods have been developed very rapidly, from analog to digital. The same goes for the orthodontic surgery. From 2008 to 2010, when I was trained, cephalometric analysis by Facebook transfer and modal surgery were routine procedures for the orthodontic surgery. However, at present, none of our residents know how to do modal surgery proficiently. It is not because orthodontic surgery is not performed as much as before, but because modal surgery is no longer essential in the preparation for orthodontic surgery. The digital work for orthodontic surgery may vary from hospital to hospital and from surgeon to surgeon. This is the preparation process I'm working on. Briefly, impression of the maxilla and mandible are taken. Final occlusion is established with models, and each is scanned three-dimensionally. The patient takes the CT scan with biting CR wax pad. It doesn't matter if it's an MDCT or CBCT. Before simulation surgery, it's necessary to properly reorient the CT scan. The surgical plan is confirmed through virtual osteotomy and simulation surgery, and the intermediate wafer is manufactured by 3D printing. In some cases, a surgical guide or mini plate customized for a patient is manufactured by 3D printing and used. Among these digital workflows, what I want to talk about today is reorientation, simulation surgery, and surgical guide mini plate. First, reorientation. In orthognathic surgery, digital work is performed based on 3D CT. However, since MBCT is taken in a spine position and CBCT is taken while leaning on the headrest, the head posture is arbitrary. Therefore, the head posture should be reoriented for accurate 3D diagnosis. Reorientation can be done in various ways, but there is no consensus as to which method is best. In 1862, Broca defined natural head position as a stable physiologic position when a man is standing and his visual axis is horizontal. In 1958, Moody Santin redefined natural head position as a standardized and reproducible orientation of the head in space when one is focusing on a distant point at eye level. Various methods for reorienting the acquired CT image to the natural head position have been suggested. I participated in the development of the skull reorientation method using the POSIT algorithm, and I will introduce this method. The pose from orthography and scanning with iteration algorithm is called POSIT for short. It's a concept that a three-dimensional object appears as a single shape when projected onto a two-dimensional plane. And this method is used in the automatic parking system of cars. To use POSIT algorithm for head reorientation, initially radioceramic markers were used. Attach 4 or more markers to the face as far as possible and take a frontal photo in natural head position. It is better if the position of the markers are different or not, anterior posteriorly. And a 3D CT was taken. The ceramic markers can be seen in the CT. And the rotation matrix of the skull can be calculated using the 3D coordinate on the CT and the 2D coordinates in the photo of the markers. If we enter the rotation matrix into the program, the skull is reoriented as follows. When the accuracy was analyzed using the skull phantom, the pitch error was the largest and averaged about 0.17 degrees. The following is how the skull is reoriented in the program. We analyzed how accurate it would be if the CT is reoriented according to the frontal and lateral photos taken in natural head position without this POSIT algorithm. And we found that the other reorientation methods are not as accurate as POSIT algorithm. I will show you a few cases where virtual surgical planning were performed using the natural head position skull reorientation method. This patient came to the hospital to correct his facial asymmetry. He showed the same eye level on the natural head position. with a picture taken according to the natural head position, we performed skull reorientation using the positive algorithm. Based on this head posture, simulation surgery proceeded as follows. The surgical plan has been confirmed. The intermediate wafer was made by 3D printing, and the operation was performed as follows. This is a photos before and 1 year after surgery. This material was well quality. For a good surgical outcome, it is important to maintain the natural head position before surgery. However, as in this cycle reported in 2016, the head tilt seen before surgery may change after surgery, especially in asymmetric patients. Therefore, it is necessary to evaluate whether the patient's natural head position is correct or not. If asymmetric patients habitually tilt their head to look more symmetrical, it is necessary to correct it before surgery. My teacher, Professor Sun Jung-hwan, referred to this as a correcting natural head position.
오늘 너무 씩씩하게 가가지고 뭐.
This patient was on hemifacial microglia patient and showed severe asymmetry, and her right eye was higher by 2.5 millimeter in the natural head position. Plagiocephaly was observed, and the shape and positions of the eye, nose, and ears were all different. So we decided to operate according to the natural head position shown by the patient. Skull reorientation was performed by positive algorithm. The surgical plan was established with conventional simulation surgery. And the orthogonal surgery was performed. These are before and 6 months after surgery. You can see that the difference in eye level was maintained. There was still a lot of volumetric differences, but the asymmetry have been improved as planned. In patients with skull asymmetry due to the congenital anomalies, natural head position-based reorientation method is considered as a good choice. This patient also came to our hospital for asymmetry correction. In the natural head position, the left eye was positioned 2 millimeters higher than the right eye. Although there was a difference in eye level, the position of the eyebrows and ears are almost the same on both sides, so I didn't think that the head was tilted. After reorientation according to the pictures, simulation surgery was done. And surgery was also performed. Pre-operative and post-operative 6 months photos. The natural head position was well maintained and asymmetry was improved. The surgical outcome of this patient was somewhat unsatisfactory. She is a class 3 patient with asymmetry, and the left eye was higher than the right eye by 3 millimeters on natural head position. Not only the eye, but also her left eyebrow and left ear are located higher than those of the right side, so I thought that the head was tilted. I wanted to set the correct natural head position with the same eye level on both sides. But the patient complained of discomfort with the change in posture. So inevitably, I performed surgery in the current head posture. reorientation, and simulation surgery. The surgical plan was confirmed like this. The midline would be moved to the right side by 0.6 millimeter. Occlusal count would be corrected by upward repositioning of the right canine by 1 millimeter. This is the surgical outcome. Although facial asymmetry was much improved, occlusal count was still observed. The problem was that from the first month after surgery, the natural head position was changing. After surgery, she showed natural head position with the same eye level on both sides. Consequently, the midline and occlusal count were reversely corrected. Although the swelling was not completely resolved yet, you can see that the right deviation of the chin and the right side looks wider. Perhaps if this head posture is maintained, I think that grinding on the right side should be performed once more. The method of head reorientation according to the natural head position using the POSIT algorithm have been described with a few cases. In order to use this method clinically, it is necessary to evaluate whether the patient's current natural head position is consistent, and especially asymmetric patients should be considered that their natural head position may be changing. If the patient's natural head position is considered to be head tilted, the corrected natural head position can be determined by considering the soft tissue, such as position of eye, eyebrows, nose, and ear. Next is simulation surgery. I have used a program called Planning for the simulation surgery, which was created through a cooperative research by Professor Won-jin Lee and Professor Sun Jung-hwan, and I was involved in the development process. For the simulation surgery, 3D model of skull and maxilla was loaded on this program. Up to 8 reference points are determined according to the surgical plan. The reference point will be used as a center of rotation and the moving point. The movement of maxilla are divided into translation and rotation. The advantage of this program is that the center of rotation and the moving point can be clearly determined. And the amount of rotation can be expressed as the distance moved by the moving point. This is the process of simulation surgery. Reference points are marked, and maxillary movement, such as panting correction, and posterior infection, can be done as planned. After maxillary movement, the location of the collision between the skull and moved maxilla can be calculated and represented as red closed curves. Recently, the entire processes from head reorientation using the POSIT algorithm, simulation surgery, to the post-operative analysis of surgical outcomes were combined using a cloud-based platform. This is a cloud-based platform in development. The advantages are that there is no need to install a simulation program on a computer. There is no need to send and receive large simulation files. Also, it is easy for surgeon and technician to give and receive feedback. The last topic is surgical guide and mini plate. This is the osteotomy guide and mini plate system I've used. Osteotomy guide is adapted to the maxilla, supported by two teeth fixed with two screws. First, drill the screw hole to fix the mini plate and mark osteotomy line and interference in the anterior wall with a piezoelectric device. After removing the osteotomy guide, root bolt-on is finished and maxilla is repositioned with an intermediate spring. The maxilla is fixed with the mini plate using pre-drilled screw holes. The method is almost similar to other guide systems. However, the difference is that both osteotomy guide and mini plate are made of titanium by 3D printing method. We use a selective laser melting method for metal 3D printing. Briefly, a workpiece is built up from a powder bed. A thin powder bed is molten up by a laser, controlled by a scanner system. When the material solidified again, a metallurgic connection is formed. The building platform is lowered. The new powder layer is delivered and the process starts again. This SRM method is known to have better accuracy than electrobeam melting method and laser metal deposition method, and can create very complex structures. By SRM method, patient-specific implants for skull, zygomatic, and mandibular angle have been usually manufactured. Recently, it was applied to the guide system for orthognathic surgery, and I used it as a clinical trial in the development processes. A clinical study was conducted to evaluate accuracy of surgery using the guide system. 22 patients with orthognathic surgery were included. For 11 patients underwent the root bolt-on surgery using 3D printed osteotomy guide, titanium plate, and intermediate spring. For the control group, the root bolt-on was performed only using 3D printed intermediate spring without this guide system. CT scan taken before surgery is T0, immediately after surgery T1, and 6 months T2. Since simulation surgery was performed in the same way in all patients, the surgical plan was calculated from the T0 CT and simulation surgery. The difference between the simulation surgery and T1 CT was calculated for the surgical error. In order to mark the same reference point in T0 and T1 CT scan, registration tool of Mimics program was used. Skull and maxilla of T1 CT were replaced with the skull and maxilla STL models of T0 CT respectively. In maxilla of T0, simulation surgery, and T1, 5 landmarks were marked at the same point using the STL triangle structure. And the surgical plan and surgical error were calculated at each reference point. In addition, since there are concerns about the clinical stability of the 3D print mini plate, the positional stability of the mini plates were calculated by comparing T1 and T2 CT. Here is the visual. Each reference point showed the methodical error within 0.05 millimeter. There was no significant difference in surgical plan between the two groups. In group 1 with osteotomy guide system, midline correction was carried out more accurately with statistical significance. In addition, in group 2, maxillary setback was not sufficiently performed as planned compared to the group 1. For vertical correction, there was no significant difference. The surgical error at each reference point is expressed as the root mean square error as follows. Group 1 showed significantly small surgical errors at 8 points upper 1 and upper 6 compared to the group 2. in group 1, there was a tendency to become inaccurate from anterior to posterior of the maxilla. On the other hand, in group 2, the surgical errors occurred similarly at all reference points in maxilla. There was no significant difference in post-operative stability of mini up to the 6 months. Therefore, 3D printed osteotomy guide and mini by this SRM method are able to guarantee better accuracy in midline correction and AP correction, without worrying about the stability of the metal plate during bone healing period. Of course, further study with more patients is needed. Today, among the digital works for orthodontic surgery, I introduced the method I've used for reorientation, simulation surgery, and surgical guide. In summary, for reorientation is the most important prerequisite for simulation surgery, and I mainly use the PDgf algorithm to perform reorientation according to the natural head position. Because the natural head position may be unstable, it is necessary to carefully evaluate the patient head posture before surgery and correct it if there is a head tilt. Planning a program used for simulation surgery has the advantage of allowing the surgeon to decide the center location and the moving point, so the surgeon is able to establish surgical plan more freely. Lastly, osteotomy guide produced by selective laser melting method has the advantage of being thinner and small volume than other guide systems. And using this, midline correction and AP correction could be performed more accurately. A lot of researches are needed in the future to establish a digital workflow for orthodontic surgery. Consensus on the method of skull reorientation is needed. And there is many other tasks remaining. I'd like to continue to research in this field in the future, and I'd like to share opinions with several professors. Thank you for your kind attention. Thank you very much.
Thank you, Professor Yang. I apologize for my interruption during your presentation. If you have any questions, please turn on your videos and also your audios.
Yes, may I have a question?
Yes.
Please go ahead, Professor Takahashi.
Yeah, thank you very much for giving me the this opportunity to talk about you. Actually, my question is actually you what you have presented is very, very nice, and actually I have to follow your way. And but I'm always wondering whether the natural head position is there at the beginning of the treatment, but actually the natural head position will be changing after the surgery. How can we imagine? How can we apply? How can I say? How can we change that kind of? How can we know that kind of the change after surgery, and how can we apply the predicted natural head posture after surgery? This is my question is how can we maybe my question is how can we predict the natural head position after surgery?
Thank you for your nice question. Actually, I don't know. What I do now is I give my patients some time before surgery to practice their head posture, changing their head posture before surgery for 1 month. In short, 2 weeks. I explained that I set the squatting natural head position before surgery with, in some cases, depending on the nose or depending on the eye level, I set the squatting natural head position and show it to the patient. And I usually give them some time to practice as the changes posture before surgery. If they accept the posture before surgery, I think that the head tilt can be changing. If in some patients, they did not accept the change posture with the pain or shoulder or neck.
Right.
In that cases, I accept. I think that their posture is correct. Last case, what I showed you today, she said that she has pain after infection of the head posture, but it was my mistake. And after surgery, she showed the changed head tilt after surgery. So I think that I should wait more time for her. It's not enough for her. No, I think.
Okay, thank you. Thank you very much. ありがとうございました。Thank you.
And next question, Professor
Can I ask
Mori?
your opinion? My name is Mori, oral and maxillofacial surgery, KU. So you said natural head position is changeable before and after surgery, and you show the different eye levels case. In my experience, so also natural head position is changed after surgery. Then, so after surgery immediately, so eye level is quite different. But so 6 months later or 1 year later, these eye level are corrected. I wonder, this is an adjustment by the orbicular muscle of the eyes. I wonder, but no evidence. How do you think about this?
Thank you for your nice question. I'm sorry, I don't know about exactly about the eye muscle, and it's very difficult question. So I'm sorry, I don't know.
Yeah, especially asymmetric cases, eye level and mouth level quite different after surgery. And can I ask one more question? So you presented two kinds of replacement of the LUFO1 osteotomy. One is with guide, and one is without guide. So with guide, how do you detect the interfering part? Maybe in the LUFO1 osteotomy. So pterygoid process, about the pterygoid process, there is some interfering part, but it's very difficult to predict this and catch this. How do you do that?
When I use the guide system, the interference in the pterygoid plate within the pterygoid plate cannot be expected before surgery. Of course, I can expect it with the simulation surgery, and I should remove it with the same method as the surgery without the guard system. Remove all the interference. Sorry, can I answer you?
So sorry, I can't catch you exactly.
So the in guide system, we can mark the interference on anterior maxilla only. So when although we even though I use the guide system, there are more errors in posterior part than anterior part. So I think that it is because the interference remains in the posterior part.
Okay. Thank you. Yeah.
Okay.
Yeah, may I ask you a
yes.
question?
Yes.
Yes, you know, Professor Mori is a specialist for orthographic surgery. So in contrast, my specialty is tumor surgery. But I think your wonderful digital workflow is applicable to tumor surgery, reconstruction after tumor surgery. So have you tried to prepare reconstruction plate by using your digital workflow?
Oh, sure. In Korea, by some company, they can make the reconstruct plate, but with 3D printing.
Okay, so you usually ask a company to prepare reconstruction plate.
Yeah, for the 3D printing.
Yeah. But I thought you have very wonderful digital workflow. So I think you will be able to make by your own reconstruction plate. Because it takes so long time to ask a company. In Japan, I think at the shortest 3 weeks. So we cannot wait for so long time. So that's problem for us.
The big problem is that I don't have the 3D metal printer. I don't have it. It's expensive. So in our university, there is no 3D printing for metal. I can only design 3D design for it. I should send it to the company for printing. Thank you.
Yeah. So Dean Kwang, I think Korea is one of the most advanced country, probably top runner in this area. So because Japanese Society and Korean Association have the joint symposium twice a year. You know that. And then we always behind. So I think would you mind educating our staff in your university? I think I will be happy to give us a chance to learn how to do it. Yes.
I think this question should be answered by our dean.
Yeah, Dean.
Dean, yeah.
Yeah, sure. Actually, I didn't get the last part of the your comments, but maybe we can invite you to our university, and then we discuss a lot more about oral and maxillofacial surgery and then dental education, right? Yeah.
Yeah. So I have a very good relationship with Department of Oral and Maxillofacial Surgery in Busan National University. So frequently they come to my clinic.
Oh, really?
Yeah. But I already heard Seoul National University is the best university in Korea. So I will be happy if we can have a a chance to learn from you.
Yeah, sure. I will. Yeah, definitely. I will. Yeah, arrange your visit, and then yeah, we also hope that we're gonna have a chances to visit Kyushu University and then look around the education systems. Yeah. Okay.
Thank you so much.
Thank you.
And Professor Jimmy, do you have a question?
No, it's fine.
Okay, I'm asking you because you turned on your
Oh, yeah.
video. Okay, thank you for the presentation, Professor Yang, and also the questions from Kyushu University. Thank you very much. I think we are almost there for the closing. But before the closing, as I mentioned from the starting, I asked you to let us know if you have any more questions. But since now we don't have any more on the chatting room, but if you have any questions, once more, please ask the presenters. Please feel free to ask if you have any. Okay, or I could let you know the email addresses of the presenters from SNU SD. If you are available from the Kyushu University, Professor Yoshizaki and Professor Wu, would you like, if you are available, could you let us know your email addresses on the chat room? Thank you very much.
Yeah.
Thank you.
I will.
I'll hand over my closing. I'll hand over my closing remarks to the Professor Young-Suk Park, who is the organizer of this SNU Kyushu Joint Symposium.
Me?
Yes, please. I know you are available.
Wow, what a surprise! Yeah. Okay. Yeah, I was very happy and honored to enjoy this wonderful lecture today. Tooth development, I might guess, by Yoshizaki Sensei, and P. gingivalis and Alzheimer relationship by Wu Sensei, and your portfolio by our Park Sensei, not me, and the 3D orthodontic surgery by Yang Sensei. They were all wonderful topics, and there might be challenges for us to thrive on and finally find a satisfactory solution as one part of future dentistry. In fact, I didn't have the opportunity to introduce earlier today, but recently my school also received a focused research center funds from the government, and it will mainly study dental materials and medical device. The head of the center are attending this symposium, Professor Liang. He might have a chance to introduce the center briefly after my talk if he's still here with us. I don't know. And I look forward to continuing this academic exchange in the future. And I hope that there will be opportunities for joint research if possible. Although it is regrettable that we could not meet directly at Seoul or at Fukuoka, we hope to meet face-to-face soon and build a friendship more firmly. 早い時間うちに互いに訪問することができることを心からお祈りします。またお会いできる日までお語りの大切になさってください。Thank you very much. どうもありがとうございました。
Professor Yang, are you here with us? Maybe, yeah. He's washing his hands. So yeah, I think the a comment from Dean Nakamura will end our session, right? Professor Chung, is it okay for you?
Yeah, of course, of course.
Okay, yeah. Thank you very much for all the participants and then the speakers. Yeah, I hope, as I already said, yeah, the exchange will continue and then future development will be done. Okay, thank you all. And then Professor, Dean Nakamura.
Yes, I believe this symposium is the second one between SNU and KYUSHU University. And then next year, I think KYUSHU University will organize this symposium. So can we request again?
Sure, yeah.
Sure.
Yeah,
Our pleasure. Yeah.
Yeah, I'd like to continue this kind of symposium. So probably both of us have many topics.
Right, exactly, right.
So I'm looking forward to seeing you next year at this symposium. Yes.
Thank you very much.
Okay, thank you very much. And maybe see you next time.
Yeah, see you.
Okay, see you. Yeah,
see you.
See you.
I enjoy so much.
Thank you very much.
Thank you, Professor Jimmy.
Maybe I think it looks like I have to organize next year.
Oh, I thought so, isn't it? I was expecting it.
Okay. Okay, see you next year.
Okay, thank you very much.
Thank you very much. See you.
See you.